So welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effect seminar series. My name is Diego, and I'm a member of the VES organizing committee, along with Stephen Airwood, Medea Soma, Yano Budu, and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alice Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. As a reminder, we're always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I just have a couple housekeeping items to mention. Each talk is about 20 to 25 minutes, though there's just one speaker today, so the presentation is going to be a little bit longer. There should still be time for some questions, so please post those questions at any time to the Zoom Q&A, and we'll read them to the speaker at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can watch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. You don't have to worry about copying this URL. I'm going to post all these links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. And for the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have a fantastic speaker scheduled for today's presentation. Nicholas Liu is a graduate student in Harvard University's Chemical Biology PhD program working in the lab of Professor Brian Liao. Nicholas's work focuses on applying interdisciplinary approaches, including genome editing and protein biochemistry, to study DNA methyltransferase 3A, which is a critical cancer target. So Nicholas, feel free to start sharing your screen and go ahead and start whenever you are ready.
All right, great. Can you see my screen or?
Yep.
Awesome, perfect. Well, thank you for the introduction and for the speakers for giving me the chance to speak today. I'm really excited to tell you about some of the work we've been doing. So yeah, so my name is Nick Liu. I am a 6th-year graduate student in Brian Liao's lab at Harvard University. I'm excited to tell you a little bit about what we've been doing studying DNMT3A using, as Diego said, a mixture of genetic screening as well as protein biochemistry. And I just apologize up front for my voice. I'm getting over a cold from a few weeks ago. So as an outline for the talk today, I'm going to start off by giving an overview of DNA methylation and DNMT3A in biology. I'm then going to talk a little bit about this screen that we did, combining a reporter system as well as base editing mutagenesis. And then I'm going to focus, for the last part of my talk, I'm going to focus on some of the characterization we did with respect to hits we found in a histone reader domain, the PWWP domain. So to start off, our lab is really interested in chromatin regulation, in particular, the ways that readers and writers of modifications to histones and DNA, how they are involved in regulating gene expression. Many of these factors are mutated in disease. And one of the most well-studied of these, chromatin modification systems is DNA methylation, which predominantly occurs in the 5' position of cytosine. And DNA methylation has a lot of different functions, but probably most well-known is its ability to silence genes. So this especially happens when it's found at promoter regions in what are called CpG islands, which are regions of DNA that are enriched in CpG dinucleotides, which are the target of DNA methylation. And so when these CpG islands are unmethylated, their genes are on, Whereas when they get methylated in these promoter regions, that actually represses transcription and silences genes. Now, DNA methylation is dynamic, and there's a host of enzymes that both deposit it and also remove it. So the protein I'll be talking about today, DNMT3A, is what's called a de novo methyltransferase. It goes into new regions of the genome and deposits DNA methylation. But there's also other machinery to both maintain it across DNA replication, as well as remove DNA methylations from positions where it already is occurring. And DNA methylation and DNMT3 in particular is really important for development. So if you knock out DNMT3 in mice, they actually become runted and die shortly after birth. And additionally, mutations in DNMT3 have been associated with different genetic disorders. For example, here I'm showing a dwarfism disorder. There's also mutations that cause overgrowth. And even apart from its role in genetics, recent work has really highlighted a role in hematopoiesis. And so if we take a look at the protein and we highlight mutations that have been found in acute myeloid leukemia, which is a blood cancer, we see that actually a very broad swath of residues have been found to be mutated. This is from the COSMIC database, really indicating a tumor suppressive role of DNMT3A. And people have shown that this gene is mutated early in leukemogenesis, and that's also reflected by the fact that it's very highly mutated in this condition called clonal hematopoiesis, which is associated with aging and occurs when you have overrepresentation of certain blood cell lineages that have gained a mutation in one of these cancer-associated genes like DNMT3A. So it's not cancer itself, but it is a stepping stone to cancer, and it's also been associated with cardiovascular disease and things like that. And so given this sort of very broad array of loss of function mutations, you might expect that the activity and the function of this protein is very tightly regulated, and indeed that's the case. The methyltransferase domain, for example, even beyond catalyzing methylation, it actually also mediates protein-protein interactions, both with other copies of itself, as you can see here, or with accessory proteins like DNMT3L. And actually, that'll be important later in my talk. But these interactions help shape its activity. The other two domains, they mediate targeting of this protein to different places in the genome. So the ADD domain actually participates in this really interesting autoinhibitory mechanism where it blocks substrate binding by the methyltransferase domain and autoinhibits it. And only when the protein encounters H3 that's unmethylated at lysine 4 does it relieve that autoinhibition. So it's a way of targeting to H3K4 unmethylated regions. The PWWP domain, by contrast, actually recognizes a different mark, H3K36 ME2, which is a mark of intergenic regions, and is also, in that way, a targeting module. But something that I want to point out is that to date, there's actually no structure that has resolved the entire protein. So what we know of DNMT3 is really built out of different pieces that we've kind of stitched together. We don't really have an understanding of how the PWWP domain contacts the rest of the protein. And crucially, given the really broad array of mutations that have been identified in disease, we don't have a great understanding of, or we don't have a comprehensive understanding of what these mutations might be doing. So that leads me to this sort of idea for endogenous mutagenic screening approach, which our lab and others have applied to study a variety of different chromatin regulators and other proteins. For example, here I'm showing just a picture of a previous study from our lab studying LSD1, which is another chromatin regulator, and specifically finding drug resistance alleles. But we hypothesized that using a similar sort of screening approach, we might be able to find sort of important residues, functionally important residues, functional hotspots, and learn about what parts of the protein are important for its function, and thereby gain insights into, where future studies might want to target the protein therapeutically, gain insights into how some of these mutations that have been observed in the clinic might be working. So that leads me to the second part of my talk, talking about this base editor scanning screen that we did. And there's sort of two components to this screening approach. One is the way that we generate mutational diversity, and the second is the way that we select for different mutations based on the function. So I'll start off by talking about the mutations themselves. At the time that we started this project, the standard way of doing these sort of CRISPR-based screens was to use Cas9 mutagenesis. And as you all may know, the way that Cas9 mutates is it actually causes a double-stranded break in the DNA, which is then repaired through processes like non-homologous end joining, which creates a diverse spectrum of indels, many of which are frame shifting. And that's actually a problem for us because DNMT3A is non-essential in cultured cells. And so all of these frame shifting alleles will just sort of like not be selected out. They'll sort of sit there in our population of cells and potentially obscure interesting mutations, because you can imagine frame shifts will be kind of indiscriminately knockouts, no matter where in the protein they occur. So for that reason, we decided to implement cytosine base editors, which if you're not familiar with this technology, it was developed in the lab of David Liu, also here at Harvard. And it basically uses Cas9 and guide RNA programmable targeting to induce substitutions of cytosine to uracil, which is then read as thymine. And you can see here, this is a little cartoon showing, one way that this might happen, where the editing is generally restricted to a window, like a 5 nucleotide window, although that can be flexible. And what you can see here is that you can essentially mutate these cytosines to thymines and substitute amino acid without generating indels. So that's good. The second problem, however, is that we needed a selection strategy to read out the activity of endogenous protein. And to do that, we adapted a reporter system that was previously developed by the Elowitz lab at Caltech. Here in these cells, we have integrated a fluorescent reporter. So this is citrine, which is essentially like a yellow fluorescent protein. And the idea is when we add doxycycline, we can actually induce the recruitment of DNMT3L, which you might remember is that accessory protein I mentioned earlier. And what that can do is recruit endogenous DNMT3A to this reporter, where upon methylation of the promoter can silence citrine expression and turn the cells dark. Now, one crucial aspect of this is that this strategy doesn't involve overexpressing DNMT3A. We can actually recruit and measure the activity state of the endogenous DNMT3A that we mutate by genome editing. And so the way we conducted our screen is we took these reporter cells, we transduced in a pooled library of guide RNAs tiling all across the coding sequence of this gene. We treat with dox and the cells silence to varying degrees. And we use FACS to isolate either citrine positive or citrine negative cells. And, just as an example, citrine positive cells are ones that are not silencing as well as you would expect them to, and therefore should be enriched in guide RNAs that cause loss of function mutations. And so first we wanted to validate that a reporter was working properly. So when we treat these reporter cells with dox, you can see here that the percentage of citrine positive cells decreases and falls to 0 over time. And even after we remove dox, the cells stay dark, which is consistent with the heritability of DNA methylation. And when we look at the histograms of citrine fluorescence from different time points, we can see that the on and off states of the reporter are very well distinguished. Crucially, we also confirmed that there was indeed methylation in these cells. So we used targeted bisulfite sequencing to look at this. And we can see here, I know this is a complicated plot, but essentially going from the lighter circles to the darker circles is going forward in time, and you can see that there's an increase in DNA methylation over time at this reporter when we're treating with docs and recruiting DNMT3A. The next thing we wanted to do was confirm that it was indeed DNMT3A that was doing this methylation. There's actually another de novo methyltransferase in the cell, DNMT3B. However, we found that only when we knocked out DNMT3A did we protect against this silencing. So you can see here in red, when we knock out DNMT3A, the cells, many of them stay unsilenced, or they say citrine positive. And similarly, when we go in with base editing and we design a guide RNA that will introduce a premature stop codon at this tryptophan 509, therefore, causing a nonsense mutation. And when we treat cells with that and cause base editing, we see a similar kind of protective effect where the cells fail to silence as well. So that all of this together indicated that our reporter was working the way we intended to and was actually reading out the activity of the native protein. So with that, we decided to go forth and conduct this experiment. And I'm showing here the results of our base editor scanning experiment. There's a lot of data here, so I'll walk you through it. Each dot is a different guide RNA, and they're colored based off of the mutation that they're predicted to create in the coding sequence. So for example, these red dots are all predicted to cause nonsense mutations. And you can see that by and large, they are scoring positively, which indicates that they're enriched in citrine positive cells, which indicates that they are loss of function, as expected. Conversely, these orange dots are all annotated to cause silent mutations, and they're largely unchanged, which again is consistent with what you might expect for silent mutations. And we also tested other controls. So here I'm showing a bar plot where we've classified guide RNAs based off of what type of mutation they cause. And we also tested intronic guide RNAs as well as guide RNAs here that don't have any cytosines in their windows. And you can see by and large, those are all also unchanged. One thing I'll note is we do see a few of these, intronic and silent guide RNAs that do enrich. And in many of the cases that we've checked, those are likely causing editing that's kind of spilling out of the window. And so it's just kind of like unannotated editing that's causing those effects. But we were really interested, especially in these missense mutations. So we see a lot of them throughout the protein, and they have diverse effects. As kind of to validate the results of our screen, we focused on two of them to start with. So this guide RNA targeting the glutamate 756 residue, this is actually a residue that is in the catalytic site and participates in the mechanism of this enzyme. And so you can imagine it makes sense that mutating it will cause loss of function. Conversely, we have one very conspicuous hit down here that is depleted and therefore, conversely gain of function. And when we map that to the structure, we actually found that it's right at this autoinhibitory interface between the ADD and the methyltransferase domains. So again, it kind of made sense that if you mutated that, it would block that autoinhibitory mechanism and therefore activate the protein. And we validated this biochemically. So we expressed and purified these mutants, and we showed that for wild type, we see a six-fold activation of activity when we add in this H3K4ME0 peptide. You'll recall earlier I mentioned that there's this autoinhibitory mechanism, and it gets relieved when when the DNMT3A recognizes this H3K4ME0 tail. However, when we make this active site mutation, we actually see no detectable activity, confirming that it's loss of function. And conversely, the mutation at the autoinhibitory interface, the G532N mutation, is hyperactive and additionally is not further stimulated when we add this peptide, which is all consistent with a mechanism where it's basically blocking the autoinhibition. So it's kind of like already activated. And so, that these results kind of indicated to us that, our screen was working and that we were getting like real bona fide hits. Next, I'm just going to like quickly summarize some other like hits that we investigated in the ADD and methyltransferase domains. So, we observed a few different hit guide RNAs that seem to localize to this interface between the ADD and methyltransferase domains, particularly in the active conformation of the enzyme. And the crucial one here was this guide targeting glutamate 907, which when we mapped to the structure, we found actually mediated an ionic interaction across this interface with arginine 556, stabilizing that interaction. And we hypothesized that this might indicate, that this interaction is important for the function of the enzyme, which we tested biochemically. We showed that, again, here, this is just wild type, showing that it can be activated by this H3K4 mesero peptide. But interestingly, for both of these mutations, even though they didn't have huge effects on the activity of the enzyme by itself, they prevented the ability of the enzyme to be allosterically activated by this peptide, indicating that this sort of interface between these two domains is really important for the allosteric activation of the enzyme, possibly accounting for, the effects of different mutants here. And this was interesting to us because if we look at mutations that have been found in AML in the Cosmic database, there's actually a wide variety of them that map to this interface. So this is one example of, being able to identify possible functions of different mutations from the outcome of our screen. So in the last part of my talk, I wanted to focus in on some interesting hits that we found in the PWWP histone reader domain and what those might, what we found those mutations to do. So, this is a zoom in of some of the screen data that I showed earlier on the PWWP domain. And when we map these hit guide RNAs to the structure, we see they're sort of broadly distributed throughout this domain. But what was really puzzling to us is why we even see these in the first place. So you might remember that the way that we designed our screen is that we had a DNMT3L come and recruit DNMT3A kind of forcibly to this reporter. So it didn't necessarily make a lot of sense to us why mutating a histone reader, a targeting module, might cause effects in this situation. So one kind of obvious possibility that we considered first was maybe these hits are just destabilizing the protein. And indeed, a previous study has shown that mutations in the PWWP domain are especially likely to destabilize the protein. So here, over half of the ones they tested were destabilizing. And we initially tested this by conducting Western blot assays to look at DNMT3A levels after we've introduced base editing mutations. We found, for example, that, one guide, this G293E294 guide, did obviously reduce protein levels, indicating that it was unstable. However, we didn't obviously see effects in many of these guide RNAs, and it's important that I note that DNMT3A is actually very lowly expressed in this reporter cell line. And so it makes blotting for it accurately very challenging. And so we wanted a more quantitative way to assess protein stability. And we turned to this fluorescent reporter approach, which was initially developed by Ben Ebert's lab. And here what we do is we express different mutants of DNMT3A as GFP fusions. And we also have mCherry expressed as a co-transcriptional control. So the idea here is that, the fluorescence of GFP is linked to DNMT3A. So if the DNMT3A is, unstable or gets degraded, it'll drag down the GFP, and the ratio of GFP to mCherry will go down. As controls, we tested a few different clinical mutations that have been investigated in the past. Both of these mutations in red have been shown to be unstable, and indeed we find that they reduce protein levels, or the levels as reported by this reporter, to about a quarter of wild type levels. Conversely, this purple mutation, the W330R, which will become relevant later, this was shown to be stable, and here we show that it preserves about 60% of expression. When we test the mutations that are caused by our guide RNAs, we see a diverse effects. So this double mutant that I mentioned earlier, we show that indeed it is unstable, but we also have stable mutations. So this R301W and this E342K mutation both seem to be stable in this assay. And we validated this biochemically as well. So we expressed and purified the PWWP domain by itself in isolation. And we looked at the thermal stability by differential scanning fluorimetry. And here I'm showing that, these two of these mutations, which were unstable in our cellular assay, are also unstable as reflected by their melting curve shifting to the left, indicating that they melt at lower temperatures compared to wild type in gray. Conversely, when we test the two stable mutants, we see that indeed their melting curves are very similar to wild type. So given all this information, we were able to assign functions to some of these mutations as being destabilizing, but that left the question open as to what these two mutations are doing, R301W and E342K. So the next step for us was to just see what happens to activity when we measure the activity of these mutants. And interestingly, the R301W mutant, or as expected, the R301W mutant is less active. We also tested this W330R, which I mentioned earlier and is in fact a mutation at the histone binding pocket. So we included this as a control because it kind of messes up the canonical function, the histone targeting function of this domain. But interestingly, both this mutation as well as the E342K mutation in vitro were hyperactive. And I'm not going to, and this is just summarizing what I just said. I'm not going to get into this too much, but we were a little confused why the E342K mutant here was hyperactive, given that it was loss of function in our screen. And so we did some follow-up genotyping analysis, and what that suggested to us is that in the screen, the guide RNA that we annotated as E342K actually also caused some bystander silent mutations, which most likely altered codon usage in a way that impaired expression. And so we believe that that accounts for why it was loss of function in the screen. But given this sort of like biochemical finding that it's hyperactive, which contrasted with the R301W mutant, we were kind of interested in following up on this a little bit more. So the next thing we did was we tested for H3K36 Me2 binding, which a proxy for which is stimulation, because people have actually shown that if you incubate the protein with the H3K36 Me2 peptide, that it'll actually stimulate the activity. So we confirmed this with wild type, where when we add the peptide only in the Me2 state, but not the Me0 state, we see activation. And conversely, as a control, the W330R mutation I mentioned it can't bind to this mark anymore, and so it's not activated. However, the R301W mutant is still activated, and the E342K mutant is no longer activated, but it's sort of like already pre-stimulated because it starts off at activity similar to the activated wild type. So given these opposing effects between the R301W and the E342K mutants, we were kind of wondering what is going on with these mutations. Why are they causing these changes in activity? When we look at the structure, we see that these two mutations are kind of along the same face of the domain, and they're on the surface. And when we align the structure of this domain to another PWWP domain containing protein that people have solved the structure with the nucleosome, when we align these two structures, we see that these mutations seem to be or are likely at the DNA contacting interface. And so we hypothesize that perhaps the effects on function might be explained by altered DNA binding. So, and I should mention that people in the past have shown that the PWWP domain binds DNA, but what was not known was whether that actually had any beneficial role for function. So if anything, people have suggested that it might inhibit the protein through substrate inhibition. But our results here suggested that possibly there was some functional role going on. So to confirm this, we initially just evaluated binding of purified PWWP domains to a DNA probe. You can see here in this gel shift assay that when we add in wild type protein, we see a shift towards these higher molecular weight complexes indicating binding. And kind of similarly, when we look at fluorescence polarization signal, we see an increase in signal as we add more protein, indicating binding. However, when we introduce this R301W mutation, which is predicted to block DNA binding because it's getting rid of a positive charge that will interact with the negative backbone of DNA, we see essentially loss of DNA binding and kind of a flat FP curve. When we test the other two mutations that have kind of opposite changes in charge, we actually see greater DNA binding and actually like a shift to the left in the FP curve, indicating again greater DNA binding. So these results indicated that these mutations do in fact perturb DNA binding, but they don't show that this effect is actually what's causing the changes in catalytic activity. So to look into that, we measured how the activity of the protein responds to ionic strength. So just to walk you through here, what we did was we incubated wild type DNMT3A with DNA substrate under increasing concentrations of sodium chloride. And the idea here was that at high concentrations of sodium chloride, the increased ionic strength will interfere with the ionic interactions with DNA and therefore make the enzyme work less well. And we hypothesized that, you know, if the interaction with DNA is weakened, perhaps that would sensitize the protein to this effect. And indeed, when we look at the R301W mutant, which is again that mutant that bound DNA less well, we see that it is sensitized to increasing ionic strength. So even at 100 millimolar, the activity falls proportionally by a lot. Whereas conversely, the other two mutations almost seem to protect against this. So they are just as active, if not more active, at the intermediate concentration, and even at the highest concentration, they remain more active than wild type. So these results suggested to us that the altered PWWP domain DNA binding affinity likely underlied the changes in catalytic activity. But one question that remained was, you know, is this effect just specific to those two mutations that we just kind of pulled out of our base editing screen, or is it general? So looking back at this interface, we identified two other residues that are positively charged and likely interact with DNA or promote interaction with DNA. And this is again, just the activity of the two mutations I mentioned earlier. When we take these new mutations and we mutate them to get rid of those interactions, we see that they are also loss of function. So this indicates that, that this DNA binding interaction is kind of broadly important for protein function. And I want to note that, you know, 3 out of 4 of these mutations have been found in different diseases such as cancer, which kind of indicates that this loss of function mechanism is relevant for DNMT3A's role in disease. Now, you know, we've shown a lot of, so previously we investigated all of these interactions in vitro, but you can imagine that, you know, DNMT3A in its native cellular context, it could be the case that this is just a biochemical artifact, and we wanted to verify that this is actually important in cells. So to do this, we measured chromatin association using this assay where we expressed the proteins in cells, we isolated nuclei, and then similar to before, we actually treat these nuclei with increasing concentrations of NaCl. And the idea here was to elute proteins by disrupting their interactions with their charge interactions with chromatin. And the idea is that if we had a mutation that impaired chromatin association, it should elute at a lower sodium chloride level. So for wild type, we see this pattern where it seems to elute at an intermediate concentration of NaCl, However, for the R301W mutant, which binds DNA less well, we see a shift over to lower concentrations as expected. Similarly, for those other two mutants I mentioned that are also impaired, predicted to be impaired DNA binding, we also see less elution at lower concentrations of NaCl. By contrast, the W330R mutant, which previously I showed was bound DNA better, is just as good as wild type in cells. Whereas the E342K mutant, we actually in repeats of this experiment, we never saw any elution, which we interpreted as maybe it is binding to chromatin really strongly and we can't pull it off. So finally, we wanted to investigate whether the effect of this altered DNA binding on activity was relevant in a methylation context in cells. And so for this, we're very fortunate to receive these triple knockout embryonic stem cells from Professor Alex Meissner. These are embryonic stem cells that have been triple knocked out for all of the DNMTs. So they do not contain any methylation, and they actually can't maintain methylation across cell divisions at all. So they're severely hypomethylated. So what we did was we overexpressed different variants of DNMT3A in these cells. And the idea here is that the DNMT3A will go throughout the genome and methylate it, and we can use bisulfite sequencing to figure out where that methylation goes and how much of it there is, and in particular with very high stringency, because the cells can't maintain the methylation. So we're only detecting kind of methylation that is being actively deposited. So when we did this experiment, we did the bisulfite sequencing, and what we found was that this R297W mutant, which corresponds to the R301W mutant I mentioned earlier, just mouse versus human, we actually saw that globally there was less methylation. This mutant caused less methylation globally than wild type, consistent with it being loss of function. And this is further confirmed by analysis of differentially methylated regions, where we showed that predominantly this mutant caused hypomethylated DMRs, whereas the other mutants were more sort of half and half. And as a control, we also tested this mutation you might remember from the beginning of my talk that was totally catalytically dead. And this was obviously very hypomethylated. So the last thing we wanted to do was see whether these mutations, as we showed in vitro, whether they preserve H3K36 targeting, which is the canonical function of the PWWP domain. As a control, we have this W326R mutation, which is the W330R in human that I mentioned earlier. And this has a mutation at the binding pocket. And correspondingly, what we see here, so what we've done is we've broken up the genome into different bins and then ranked them based off of how much H3K36Me2 there is into quartiles. So you can see that whether you're at high H3K36Me2 or low K36Me2 regions, there's an equivalent amount of methylation, indicating that this mutant has no preference. However, for wild type, for R297W, and for E338K, all of these mutants show increasing methylation at regions of the genome that have higher K36Me2, indicating that even though they perturb DNA binding, they still preserve the histone reader function. And we can see this in finer detail if we break up, if we classify these pins not into quartiles, but into 1,000 quantiles. With wild type protein, we can see a very clear increase in the higher amount, the more H3K36Me2 there is, the more methylation there is. Conversely, when we have catalytically dead, we see no methylation. When we have no binding to K36, we see no preference. But for the other two mutations, we do see an increasing trend. And so just to summarize, like all of this brought us to a model where the DNA binding, even beyond recognizing this H3K36Me2 histone mark, the DNA binding role of the PWWP domain is also important for activity of the enzyme. And when we mutate through base editing or, perhaps like with clinically observed mutations, when we disrupt this DNA binding interaction, this actually causes impaired methylation, which we show does occur also in cells. So if you're interested in reading more, and there's a lot of details I skipped out, I wasn't able to talk about due to time, I encourage you to check out our recent publication in Nature Chem Bio. And just as a summary of what I've told you, we applied mutational scanning empowered by base editing and endogenous methylation reporter to essentially systematically identify loss of function mutations throughout the whole protein, including many cancer-associated ones. And through kind of deep mechanistic biochemical investigation, we showed that, in fact, there is a non-canonical function of the PWWP domain, namely its ability to bind DNA, that is necessary for full activity. So with that, I just wanted to acknowledge a lot of people that helped make this work possible. So Brian has been a fantastic mentor to me in the lab and has allowed me to work on this. I also want to thank Emma Garcia, who is a very talented graduate student that I've worked very closely with on this project and on other projects. She contributed a lot to this work. So I really wanted to give a shout out to her, as well as many other members of the lab that have provided helpful advice, as well as coding expertise. I want to thank John Dench, who at the Broad Institute, who is our collaborator and helped contribute the base editor plasmid, as well as helped us design our library. David Baldock at H3 Biomedicine, who is an expert in biochemistry, DNMT biochemistry, and was a really invaluable source of advice, as well as Alex Meissner, who I mentioned previously, contributed the triple knockout ESCs to us. And with that, I'd like to again thank the lab as well as funding sources, and thank you for listening, and I'd be happy to take any questions that people might have.
Thank you, Nick, for such a great talk. We have time for questions now. So if I'm looking at the Q&A. Okay, I think I can start with one of my own.
Mm-hmm.
So I loved the methylation reporter that you presented or that you adapted. Have you considered using this system for other methylation editors, or is this for future directions?
Yeah, that's a really great question. So one of the, I think, advantages of this approach is that it is generalizable. You can imagine fusing other recruitment mechanisms or other chromatin modifiers. And that's definitely something that, yes, we've sort of left open for future work. One thing kind of addressing that question is you might notice that something that we have looked at, if I go all the way back, something that we have looked at is actually alternate recruitment mechanisms of DNMT3A. So I mentioned that DNMT3A also interacts with other copies of itself. So something that we've investigated in a separate project is like whether we can observe different enrichment profiles when we recruit DNMT3A through alternative mechanisms. So that's something that we are hoping, is really being led by Emma Garcia, the other graduate student I mentioned. And that's something that we're hoping to share in the future. But yes, certainly, like this reporter system can definitely be adapted to studying other DNA methyltransferases, as well as other, even histone modifications that are, silencing for that are gene silencing. Yeah.
We have a question. So first of all, great talk, Nick, from Matthew Coelho. The question is, how do you deliver the base editor to cells?
Oh, yeah, that's a great question. So we deliver the base editor through lentiviral transduction. So we have a, and these plasmids have been deposited by John Dench, our collaborator, onto Agene. They're essentially like one plasmid systems that express the guide RNA and the base editor together. And you can integrate them into the genome through lentivirus. So when we perform these screens, if you're familiar with CRISPR screening, what you typically do is have a pooled library and then you transduce at a low multiplicity of infections. So each cell receives one guide RNA, and then you can basically mutagenize in parallel in a pooled fashion.
There is a follow-up. Did you look at DNA mutations caused by base editing, or how did you do it?
Yeah, so that's also a really good question. And I kind of glossed over a lot of the genotyping that we did in this talk. So we did confirm for many of the mutations I mentioned, for example, the kind of the control mutations, the active site, and the autoinhibitory mutation, as well as those PWWP mutations I mentioned. We did do genotyping to confirm that there were C to T edits in the expected positions. Something that we did find, which is, I think, important for future base editor screening papers and is also consistent with what a lot of other groups have found, is that with base editing, you also oftentimes will get editing that bleeds outside of what you might annotate as the editing window. You know, you might also get bystander mutations, like, for example, silent mutations. And it is important to also consider what effects those might have. So I mentioned earlier in my talk that we saw this discrepancy between the E342K guide, which was loss of function, and the mutant itself, which we showed biochemically was overactive. And that we found was likely due to these bystander passenger mutations. So yeah, that's a really great question. And we did look into a lot of the genotyping. And so there's definitely a lot more detail on that in the paper.
Okay. So we have comment, great work. You did a lot of interesting biophysics for this paper. Are there other questions in the chat? Or there we go. Okay, I think if there is nothing else, I think we can end it here. So thanks everyone for attending. Just as a reminder, we have this seminar recorded and it will be posted on our YouTube channel. So you can tune there to re-watch it. That's it. Thank you again, Nick, and have a good rest of the day, everybody. Bye.
Thank you.